Today we're going to be continuing with chapter 1 on important biomolecules and nomenclature. In case you missed it, last time we went over what is microbiology, a general overview of the subject, went through what is life, what makes something living, non-living, and a brief history of what brought us to here, talking about ideas of spontaneous generation, how do we sterilize things and make things safer for patients. So without further ado, we're going to get into microbiology and try not to be like Roger here, who's cramming for his itsy bitsy tiny microbiology midterm with his tiny textbook. Okay, so important biomolecules and nomenclature. Okay, so important biomolecules is where we're going to start. And the first thing we need to know are macromolecules. So we have macromolecules, or large molecular compounds that are assembled from smaller subunits. So each of the types of biomolecules that we're going to discuss today have monomers, or simple biomolecules that can be linked together. And these monomers are going to get made into polymers. So strung together, kind of maybe think beads on a string or Legos put together to make a bigger molecule. And these polymers will then be put together to make macromolecular complexes. So before we get into that, I had a little pre-quiz for you all. So you can pause me if you want to and check and see if you can think of these. We have 4 main classes of biomolecules. When we talk about life, see if you can think of what those 4 main would be. See if you can describe them, maybe what they look like, their structure, what they do, and where you might find them. All right, so if you checked in, we're going to go on and check your answers to see if we got all 4. Don't worry if you didn't get them. You can check back again at the end of the lecture and see if you have it then. So our 4 main macromolecules that you might find in a cell are lipids, carbohydrates, proteins, and nucleic acids. Okay, so we're going to go through each of these, but a brief overview. Lipids are going to have fatty acid tails, and we're going to have polar and non-polar components to them. You'd find them maybe in the cell membrane, the phospholipid bilayer. Carbohydrates, these are sugars. So when we're talking about having monomers and polymers, the monomer would be something like a glucose molecule, and then we could string them all together to get things like chitin. Okay, that might be found in like the outside exoskeleton of an insect. Okay. We have proteins here. So these are also made from small subunits. We have amino acids here, and with the little like light blue part here, we have a valine, okay? And we string them each together and we make a polypeptide that can then get folded up and have its own function in the cell, whether it's for structure, maybe it's making a ribosome or an enzyme that's important for the cell. And then our last one, nucleic acids. This would be DNA or RNA. Okay, so these are going to be made of sugars and phosphates bound together here. We have a double-stranded version, that would be DNA held together with hydrogen bonds. And we have our single-stranded version with our phosphate, our sugar, and our bases. And the single-stranded one is known as RNA. Okay, so diving in a little bit deeper, we're going to start first with carbohydrates or sugars. Okay, so sugars come in 3 main varieties. You have monosaccharides, which, as the prefix mono here suggests, are the simplest. They're going to be made of 1 carbon sugar ring. So 2 examples of that would be glucose and fructose. Okay, so if we go back here a slide, they would be these monosaccharide rings. They have carbon rings with an oxygen and maybe some extra groups out here, but they're going to be the simplest. The next one, kind of the middle one, is the disaccharides. These are sugars composed of two monosaccharides. So if we put together a glucose and a fructose, then together they would make a disaccharide. And we have three different varieties. You can have a lactose, maltose, sucrose. There's more, but these are the most common disaccharides that you might see. And polysaccharides are polymers where you string together 5 or more monosaccharides. So if you have a glucose, 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 oh, that would be a polysaccharide. Okay. Carbohydrates are compounds primarily composed of carbon, hydrogen, and oxygen. They make that ring structure, and in layman's terms, we would call them a sugar. They can be 6 or 5-membered sugars. So hexose, a hexagon has 6 carbons in the circle. A pentose or a pentagon 5-carbon sugar would be something like ribose. Polysaccharides contribute to structural support within the cell. They offer a layer of protection for the organism, and they also serve as nutrients and energy storage. So if you think back to Bio 1, when we talk about like glycolysis, the electron transport chain, glucose or a monosaccharide would be one of the things serving as that nutrient. Some other more complex versions or varieties of polysaccharides are cellulose. This is a long filamentous sort of polysaccharide. You would see this one in a plant cell wall. Agar is another polysaccharide. This one is found primarily in seaweed. And the reason we're differentiating this is we use agar to make media to grow bacteria on, or you have little petri dishes. We'll see them in chapter 2. Okay, but plastic petri dishes that we make a gelatin kind of little thick jello layer out of agar, and then the bacteria can grow on it. Chitin is a polysaccharide found in the exoskeleton of fungi. You'll also see peptidoglycan. This one comes up a lot when we talk about bacterial cell walls. It's a network of polysaccharides, these zigzagging kind of sugar molecules that can crisscross or cross-link to form bacterial cell walls and give it that more rigid structure. Again, because bacteria don't have a skeleton like ourselves, they require very stiff cell walls to give structural support. The glycocalyx is a final variety of polysaccharides here. It forms a filamentous network of sugars that forms a coat on cells. Okay, so you'll talk about later the glycocalyx or slime layer, kind of a capsule outside of the bacteria. And this acts more as protection for bacteria. It makes them harder for our immune system to get rid of them. Okay, our next biomolecule, lipids, are composed of fats, phospholipids, steroids, and waxes. Okay, so 2 key vocab words are triglycerides, tri meaning 3, and then they have a glycerol molecule. Okay, so these are storage lipids that have a single molecule of glycerol bound to 3 fatty acids here. Okay, so we have pictures here where we take our 1 glycerol here in pink and we plug in multiple lipids on the end of it. Triglycerides often comes up when we talk about like fats and nutrition. Glycerol is a 3-carbon alcohol. So we have our 3 here, and then here's the alcohol OH group on each. We'll often see when we discuss fatty acids that they can be saturated or unsaturated. So you see saturated here has all single bonds. There's no more room, no more electrons to share, to add any more hydrogens. Whereas our unsaturated has a kink in it here because, again, we have double bonds, which means if we broke the double bond, we could add more molecules to it. It's not saturated, not filled up. Fats are often discussed when we talk about membrane lipids. Okay, so the membrane is composed of the phospholipid bilayer. Okay, so we have two sets of phospholipids to form a layer made of two of them here. So, and they're all lined up so that the polar head that is hydrophilic or water loving is facing the water on the outside, and the hydrophobic water hating non-polar tails are facing each other in the middle here. And they can have saturated or unsaturated tails. They can be different lengths, and these can change how fluid the membrane is. You also notice from this picture that it's not just phospholipids, but in the membrane, we can have different proteins or transporters inserted in between here. You'll also notice if we zoom in a little further, we have our proteins inserted in the phospholipid bilayer. There are also these small brown molecules. And if we zoom in a little more, we can see that there are these ringed structures with a carbon tail known as cholesterol. Okay, so steroids are complex ring structures commonly found in the cell membranes and in hormones. Okay, so they have a ring structure and they have a carbon hydrogen tail out here. These could be cholesterols, they could be estrogen, testosterone, a wide variety of different hormones that could act as fats or as signaling molecules. Cholesterols are commonly found in the cell membrane, help with signaling, and they also change the fluidity of the phospholipid bilayer. Our third category today, proteins, are predominantly organic molecules that form long chains and can get folded in a variety of manners to perform work within the cell. They're composed of amino acids, which are their monomers or their building blocks. There are 20 main amino acids that occur naturally. They're shown over here. And based on which amino acids are a part of the protein, you get different chemical and structural characteristics. Okay, so you can see over here, some of them are non-polar, some meaning they'd be avoiding water, polar, they're going to be attracted to water. Some are charged. So as you can imagine, the positively charged ones might want to fold to interact with the negatively charged ones. Some are neutral. And based on their characteristics, we get different folding patterns, different binding pockets, and different structures. You'll often hear that structure determines function. So based on which amino acids are in what order can determine the structure, and based on its structure, it will tell us whether it's an enzyme or whether it can survive in high heat or low heat, or if it's very big or very small, can it catalyze things, all based on its structure. A peptide is a molecule composed of short chains of amino acids. So maybe I strung together 3 or 4 of these over here. Whereas a polypeptide is usually at least 20 amino acids long. These amino acid chains get folded in a variety of fashion. So first, we start off with the primary structure. So this would be the linear structure, the sequence of amino acids that we go, okay, Tryptophan is next to lysine, which is next to histidine, which is next to valine. Okay, so how are they, the beads on the string, strung together? And then from here, they get folded into their secondary structure, which could be if they're forming a helix or kind of this looped structure, if it's kind of random, or if they're kind of folding sheets, beta pleated sheets. Then these secondary structures get further folded up into their tertiary structure, where you can see like the alpha helices is coiled up really tightly, and then it gets folded a little bit more. Again, trying to go for that complex structure that will determine function. And then this final level of structure, the quaternary structure, is when multiple polypeptide chains interact together. So this one, the green one, was a linear structure here, and the purple one was its own linear structure, and they all get folded up together to perform their job. Different sequences will get different shapes. So as I mentioned, based on charge, or whether it's polar or non-polar, if it's really big or really small, can determine their shape. The structure that they form will determine their job or their function within the cell. Proteins can react only with molecules that fit their structure or their complement. Okay, so this is why when we see mutations and they affect the protein structure, they can change which molecules they can bind with. A couple of examples of proteins are enzymes. These are proteins that can act as catalysts to facilitate different reactions, to make reactions more likely to occur, expedite the process. Antibodies are also a form of protein molecules that are important for an immune response. So they'll act very specifically, binding with their antigen to invoke an immune reaction. The functional 3D form of a protein is known as its native state, or its intended, very basic, planned state. If the structure is disrupted, be it from something like a change in pH, a change in temperature, maybe it's been mutated, any of these disruptions would be said to have denatured the protein. Our final category of biomolecules is nucleic acids, and your book refers to them as the cell's computer and its programs, or maybe a better way to see it is the blueprint of the cell. Nucleic acids are polymers or strands of nucleic acids. They can form either DNA or RNA. DNA, shown here on the left, is the double-stranded variety. And you'll see there are phosphates, we have our sugars, and our nitrogenous bases are shown in the like pink, brown, green, and purple. There are 4 main nitrogenous bases in DNA, A, T, C, and G. And they're held together in the middle of the helix with hydrogen bonds. When we go look at RNA, it looks very similar. We still have our phosphate. It has a slightly different sugar. Instead of having deoxyribose, we have a ribose sugar over here. And we have similar bases, except for this time, we have a uracil instead of a thymine. This leads us to the idea of the central dogma, which was mentioned in our previous lecture, but to reiterate, the central dogma is the flow of information in the cell. We go from DNA to RNA to protein. DNA is our long-term storage, that double-stranded molecule that is for keeps. It can get opened up and we can make RNA copies that we can share throughout the cell. So we'll make single-stranded RNA from our double-stranded DNA. And then this RNA can be used as a plan to make the proper proteins. So we're going from DNA to RNA to protein. There are many varieties of RNA, and they're all going to be involved in regulating gene expression and protein synthesis. Last time we talked about some of the regulatory RNAs in passing, things like short interfering RNAs, long non-coding RNAs, but the main ones that are involved in protein synthesis are mRNA or messenger RNA. This is the one that's going to be providing the code or the message to actually make the RNA. Others that are involved are tRNA or transfer RNA. This is going to be the RNA that's bringing in the amino acid. And rRNA, this would be the ribosomal RNA that helps bring the ribosomes in, forms the ribosomal catalytic site to actually make the protein. And then we have all those regulatory RNAs that I mentioned before, long non-coding, short interfering, short hairpin, microRNAs that are regulating how much protein are we making, how long is the RNA hanging out for, things like that. ATP or adenosine triphosphate is technically going to be in this category because, as you can see, adenosine is one of the bases that we would see in DNA. And it has the sugar attached to it and a phosphate. And if it was just about right there, that would be a part of the DNA. But if we had multiple phosphates on the end and get an adenosine triphosphate, we can use this molecule as the energy currency for the cell. It's often referred to as ATP. So this would be the primary source of energy when we start talking later on with metabolism, glycolysis, the electron transport chain. We'll talk about how much ATP is actually being produced. Some fundamental characteristics of the cell are that it's an individual membrane-bound entity. So when we talk about a cell, we have a cell, and it would need its individual membrane, that it's its own discrete entity. It has to be living, going back to our last lecture, being able to reproduce, do its own metabolism, react to its environment. And the cell is known as the smallest unit capable of independent existence. So we can put multiple cells together and they'll still exist. But if we take the cell apart, it's still neat. We can have enzymes that react, but they're not going to do their own thing, reproduce on their own, respond with metabolism. They don't have that functionality on their own. All cells share common characteristics, including that they're organized, they have defined shapes. You'll see in microbiology, we'll talk about the different cell shapes later on. Things like, is it a bacillus? Is it a coccus? Is it a spirillum? They have very defined shapes. They may not all be the same, but they're very organized and discrete. They have internal contents encased in a cell membrane. So again, they have that discrete outside barrier, and their internal contents is, again, very organized, whether it's eukaryotic and has membrane-bound organelles or prokaryotic, no membrane organelles. Either way, it's highly organized. All cells have genetic material. They're going to have DNA that is their blueprint of what proteins and RNAs they're going to make, what metabolism they're going to do. They have that at their core. They will all have ribosomes or the little machines that are going to make their proteins. And even the simplest cells are highly complex entities. Okay, so I would encourage you, as you're studying this, I look at this and think, how could she make a multiple choice question? Just food for thought. Maybe you go, which one of these is not something that all cells share? Or maybe you think about all cells share, oh yeah, they all have genetic material. Just something to be thinking about in the background, because all our quizzes and our exams are going to be multiple choice. So again, to reiterate here, we have carbohydrates, lipids, proteins, nucleic acids are the 4 main macromolecules you'd find in cells. Carbohydrates being sugars, whether they're monomers, dimers, peptide, okay? We have our lipids. These would be fats, oils, cholesterols, phospholipid bilayer would all fall in that category. Proteins, which would be chains of amino acids that can form enzymes or antibodies or machinery of the cell. And we have our nucleic acids, DNA, RNA, you could even include ATP that have their nitrogenous bases, the sugar, and the phosphate as a part of their structure.